So, good morning, good afternoon, and good evening. My name is Tim Devling, and I'm the Director of Sales and Applications here at Ithera Medical. And it's with great pleasure that I welcome you all here today to listen to Dr. Marty Pagel talk about MSOT and MRI, so complementary modalities for preclinical imaging. The webinar will last for about 40 minutes, and we're going to have plenty of time for questions at the end. So should you have any questions, if you look in the Go To tool, you can see a question box and type them there. We'll also be recording this webinar and we'll send you a link to this in the next few days. Marty has also very kindly offered to share his slides, so we'll also send out a link or contact us if you want a copy of those. So we welcome today Dr. Marty Pagel. Dr. Pagel has focused on molecular imaging research during the last 20 years in both industry and academia. He is a professor in the departments of Cancer Systems Imaging and Imaging Physics at the University of Texas MD Anderson Cancer Center. In addition, Dr. Pagel has held leadership positions in professional societies, funding agencies, and scientific journals that focus on molecular imaging. And Dr. Pagel's current research focus is on photoacoustic imaging, CEST MRI, DCE MRI, and PET MRI for studies in both mouse models of cancer and clinical trials with cancer patients. So over to you, Marty, and I hope you all enjoy this webinar.
Well, thank you, Tim. I really appreciate the chance for you to organize today's session because I really enjoy MSOT and MRI. It's great to have a chance to talk about both. In fact, this reminds me that this all started for me back in 2013 at the World Molecular Imaging Congress meeting when I met your colleague Tullio Giannetti at the Ithera Medical booth. And I was really impressed with the data quality coming from the MSOT instrument that he showed to me. And I was thinking, you know, an MRI person could really make use of this MSOT technology. I can see some real synergies here. And so I really got to thank Tullio for pointing me in the right direction. And well, 6.5 years later, here we are today. So it goes all the way back to just a simple conversation at a meeting, which is how often our research gets started. So I'd like to start, let's see, there we go. So I wanted to list an outline for today's presentation. Here are the 5 topics that I would like to cover. I realize the font for these references is very small, but as Tim mentioned, this presentation is being recorded and I can provide the information in these slides as well. I'll have my email at the end of the presentation, for example. So you can always look up these references later. So first, a brief introduction to multispectral optoacoustic tomography. I think many of us are familiar with the technique, but just as a brief reminder, this technology starts with a laser shining into tissue in the near infrared region of the spectrum. Molecules then absorb that near infrared light, or some molecules, and undergo thermoelastic expansion. And while not really a molecular vibration, it's more of a macroscopic effect of this expansion, which then causes ultrasound waves to form within the tissue, originating at the site of the molecule. So then ultrasound can detect those signals, and using math that's way beyond my understanding, an image then magically appears on the MSOT instrument. So I'm really glad somebody else figured out how to make the images appear, because I still don't quite understand that part. But what I do really appreciate are these advantages of MSOT. In particular, the instrument can collect an image within 1 second or less. Actually has a 100 millisecond repetition rate, but due to signal averaging, 1 second per image is pretty typical at each wavelength. And for comparison with MRI, that's really quite nice, because in MRI, it often takes us multiple seconds to collect an image, if not even longer. The spatial resolution of MSOT, about 150 micron, pretty comparable to MRI, and as a key advantage, about 100 nanomolar minimum detection level. That's a bit of an aggressive number. Certainly detection below 1 micromolar is very feasible with this technology relative to MRI, which is much less sensitive. And then an important feature of all of photoacoustic imaging, including the MSOT instrumentation, a 3 centimeter depth of view is certainly feasible. Beyond 3 centimeters, there can be some imaging and becomes a little more noisy, but I think we can confidently say we've got a 3 centimeter or 2 to 3 centimeter window into the tissue. which is great. And we can image the whole mouse then, so we can compare with MRI, small animal mouse MRI. So at MD Anderson, we're fortunate to have an MSOT Envision small animal scanner, as shown on the left. Some of these photos are provided by iThera. I really appreciate that. And on the right, you can see we also are fortunate to have an Acuity clinical scanner. And within the just next few days, we're excited to finally file our investigational device exemption with the FDA so we can start our clinical trials here at MD Anderson. So that works well. So I should point out that MSOT is really not intended to be an anatomical imaging modality. And that can be seen on the top part of the slide, where for those of us who do MRI, the view of a single frequency image might be disappointing. We're used to nice, crisp MR images of beautiful soft tissue anatomy. And similar to ultrasound, well, MSOT does give more of a fuzzy type of picture, and you have to have some experience for finding certain organs. And that's very reasonable for this technology. And because really MSOT is designed to be a molecular imaging technology, not necessarily for an anatomical reference. We have other modalities that can do that. And that's really shown in the bottom of the slide, where molecules such as oxyhemoglobin, deoxyhemoglobin, melanin, even lipids have certain spectral signatures, so that if data is taken at multiple wavelengths, as shown in the middle image here on the bottom, then the data can be spectrally unmixed, and we can literally image molecules. Now we can find those molecular locations. Again, if you just take an image at one wavelength, it doesn't look like much, and really multispectral is the key. And that's why I really appreciate that Ithera Medical decided to name their instrument a Multispectral Optoacoustic Tomography machine, really highlighting the advantage of this technology. And here's some epifluorescence validation shown on the slide as well, just to show the location of this fluorophore, or this MSOT agent. So with that in mind, let's look at some examples of how molecular imaging with MSOT can be merged with MRI. And the most obvious topic to first cover is how we're going to co-register these two modalities together, because that's important to get good quality data. Sarah Bondic at the University of Cambridge and her colleagues recently published a great paper in photoacoustics, and I'd like to walk through that paper here to show the advantages or the techniques that are needed to really improve the co-registration. So the schematic here in the upper left corner shows a typical axial image with an MSOT instrument. The mouse is wrapped in a bag and placed in a tank of water, and therefore the mouse shape has a cylindrical shape to it. In MRI, we typically place a mouse in a cradle, and the mouse body gets a little more flat. And so right away, you can see that merging these two images is going to be challenging because they're different shapes. And so a typical MSOT image and MR image are shown in the middle of this screen right now. By convention, radiological convention, MRI often is shown with the mouse somewhat upside down. I'm going to flip that around so it's easier for us to see the comparison. And then these investigators did a very nice job of contouring the body and the tumors. That's shown in this schematic here. And then these can be merged in a two-step registration process. First, by optimizing the body-to-body orientation, and then the tumor-to-tumor orientation to further refine that registration, a two-step registration. But you can see the registration is just okay, not great. Well, the key to their technology is really this idea of creating a silicone bed to hold the mouse in a more cylindrical orientation. And therefore, the images that are seen in the middle of the slide on the right really do match much better, where the tumors from MSOT and MRI clearly match better because the shape of the mouse is more similar between these modalities. Again, by contouring and then by doing a two-step registration, you can see that the tumors are merged together really very nicely from these different modalities. So the dissimilarity coefficient went up quite a lot. And that really leads me to a rather important point that I think is not emphasized enough in our field of molecular imaging. I personally think we got to spend more time handling the mice in an appropriate way, maintaining their physiology so each mouse is the same from scan to scan, or different mice on a certain day all have the same physiology, same orientation, same location within the instrument, because removing that variability of the mouse is important when we're trying to evaluate our imaging technologies. I've got enough problem with variation from the image acquisition and the image analysis, and I don't need a bunch of variable mice affecting my analyses of the equipment. And so that's why I really appreciate this example from the Bone D group, showing that with careful attention to how to place the mouse, that can really help the quality of your data. So Jan Kloos has also published a paper with his colleagues at the University of Zurich, showing a very nice co-registration of the brain, mouse brain, first by putting the landmarks on the MR images of the brain, and then also in the MSOT images, and then doing an affine rigid transformation of rotation, translation, and scaling to co-register the images. And that comes out very nicely. I think it's important to point out that they do clean up the images with some denoising, as shown in the upper right of this slide as step 1. And then they move on to doing some contouring, some rather advanced selection of the landmarks that they need to co-register. And then finally, they do the registration step. And so then to summarize this part, I think a key step here, really the second bullet item here of this list, multi-step registration process is important. Just taking 2 images and trying to slap them together doesn't always work very well. And doing it a little more carefully in 2 or 3 steps can really help. And then also this example from the Kloos group shows that refining the images before registration is also a key step, in this case denoising, and the Bone D group also has some refinement of their images prior to registration that can help. Jan's paper did point out something that I think is important for both modalities. 3D registration is often better than 2D, and that is yet to be really shown with MSOT and MRI, and would be a great application for the future. Also, deformable non-rigid registration is coming along very nicely with MRI in the clinic, and those techniques could be applied for MRI and MSOT as well. All right, so now we got 2 images that are co-registered. What can we do with that? Well, perhaps the most obvious application is to use MRI as the anatomical reference for MSOT, because as I made the point before, MSOT is really intended to be a molecular imaging technique and not necessarily to get high definition anatomy images. So here's an example from the Kloos group at Zurich again, where they were able to identify regions where this agent was collecting within the mouse brain 60 minutes after injection. And that coincided with the beta amyloid deposition as discovered through anatomical referencing with MRI. And so they could see that they're getting uptake of the agent in these amyloid deposition regions as expected. So very nice study, very high statistical significance between wild type and the amyloid beta mouse model. Now this might seem like an obvious example. Intensity from the MSOT image is very nice. You can almost guess that that's where the region in the brain should be within the MSOT image just by itself. But it's very nice to have the validation from the MRI. And I think that's really shown in this next example, also from the same research group, where they made an ischemia reperfusion model of the mouse brain. And in fact, just to jump ahead here, in this image at the very bottom of the screen, a bit on the right, you can see that the MR contrast is slightly different on the left side of the image than the right side of the image, showing that there was an ischemic insult given to this mouse. So there's something going on the left side of this image. Well, they also injected a contrast agent that has a tether between 2 chemical entities. And by causing these 2 chemical entities to be close together, it does not provide an MSOT signal. But after cleavage of MMP sense, typical agent also used for fluorescence, the 2 chemical entities can diffuse away from each other, and then they become MSOT active and can be detected. So this is a great opportunity to image enzyme activity using MSOT. And so what I think is really important to point out is that they were able to detect MMP activity using this probe and MSOT with this small green area within their ROI identified from the anatomical MRI. However, there was also a lot of green area that was outside of their brain ROI area. And so it would be somewhat challenging to know how much signal to actually quantify that is in the brain from the MSOT image. And so by using MRI as an anatomical reference, they were able to do the experiment correctly. Unfortunately, they still saw a significantly different signal uptake in their ischemic model relative to the sham, showing that they are detecting an increase in MMP activity due to ischemia. So a very nice example to show that you really do need anatomical reference, or you may not be circling the right area of your image in order to get your imaging signal. So as a third example of using MRI as an anatomical reference, Malini Olivo and her colleagues at A-STAR published a very nice study a couple of years ago looking at the evolution of adipose tissue to brown fat. And they incorporated an IRFP720 reporter gene, a protein that can be detected with MSOT, and it was only expressed when an uncoupling protein was also co-expressed in the cells. And this uncoupling protein is a biomarker of this evolution of standard adipose tissue into brown fat. So they're watching the evolution of brown fat in this graph in the lower left, where the IRFP720 signal increases, showing that this uncoupling protein is also being expressed and therefore brown fat is forming. Interestingly, they could also detect the lipid signal at the same time. And so here you can see this greenish area disappear from day 7 to day 10, or at least go down. And that can be seen in this graph in the center bottom of the screen, where the mouse model that was treated in a way that would cause the adipose tissue to turn into brown fat actually saw a decrease in lipid signal. Brown fat has less lipid content than regular adipose tissue. So very nice example from MSOT. Well, where does the MRI come into play here? Well, they did something rather clever, I thought. They performed PET MRI, where MRI was used as the anatomical reference that's shown in the center slide here with these white arrows pointing to the adipose tissue. And then by doing FDG PET, they could look at the amount of metabolism caused by glucose uptake, or as represented by glucose uptake in the PET image, a very standard methodology, of course. And so therefore, they could then relate through the MRI how much of the MSOT signal was due to this reporter gene imaging, and how much of the signal, or how much signal is there from the FDG PET, a percent injected dose measurement. And there is some correlation between the two. Not a great correlation, could be due to the biology. Also, just to show the integration of these slides, the Bone Deep group had a very nice example of how to use a silicon mold to hold the body in a cylindrical shape during MRI, and that technology could be used here perhaps to improve the correlation as well. But here's the most important point that I want to make about this slide. Perhaps MRI can be used as the fulcrum for the anatomical reference for many imaging modalities, so that MSOT can be linked with PET or with other types of modalities as well. Because as we all know, MRI has great soft tissue contrast and is great for anatomical imaging. So I thought that was a very clever multi-modality example from the Olivo group. So there aren't that many examples of MSOT and MRI that have been combined in a complementary way. Here's one example, and I'm really hoping that there'll be more examples in the future. This paper is actually available online, but the publication hasn't come out in full hard copy form yet, showing that this is relatively new to find complementary biomarkers with the two imaging methods. So in this study, and I was able to work with Harish Kaptani at the University of Liverpool, who has a great research program looking at kidney function and the effects of kidney injury and so on. And so we were able to measure the extracellular pH using MRI, a technique known as acidocyst MRI. And while I'll explain a little bit more about these graphs at the top of the screen later, I just want to point out right now the pH of the kidney tends to be low at 6.3 or so in all of the data sets that were collected and formulated in different ways. And that's fairly well known that kidney pH is relatively low compared to the rest of the body. So we have good healthy kidneys here. But then Harish and his group injected an indocyanine green contrast agent. They could watch the accumulation and wash out in the cortex as well as the pelvis, as shown in the lower left of this slide. in a very dynamic way. And that's very similar to dynamic contrast enhancement MRI, which I'll get to later as well. And so a nice example showing that MSOT can also look at dynamic properties, in this case of the kidney. Well, here's the key to the study. So the decay time, or the rate of decay of this cortex data, and then also the pelvis data, can be measured. And if MSOT is performed first, it's a nice clean kidney, normal physiology, very small amount of volume is injected intravenously into the mouse. ICG can be detected at very high sensitivity with MSOT. So you get a nice fast washout time from the cortex for this agent. However, if you perform MRI first, where we need to inject a very large amount of fluid, 200 to 250 microliters, at very high concentration, nearly 1 molar of an agent known as iopamidol, which is tends to be slightly viscous at that very high concentration. Well, that causes the kidney to take some time to clear all that MRI contrast agent, and therefore the MSOT, this is MSOT data shown in the center of the slide. The MSOT data showed that the cortex washout time was actually slow compared to the decay time when MSOT was performed first, showing that the MRI actually affected the MSOT imaging because it was performed first. By the way, the pelvis was not as affected by this measurement. And as validation, if the MSOT is performed after the MRI, performed before the MRI on day 1, and then also on day 4, you can see that the kidneys do recover from the MRI that was performed on day 1, showing that this really is an effect of the imaging modality, the MRI on the MSOT. And again, I would just like to further emphasize that we need to take the variability out of molecular imaging with regard to the mouse model. You got to think carefully about how you set up your experimental design, especially with multimodality imaging, because now you've got the mouse and these imaging instruments for even a longer period of time. And it's important to think of how the mouse is responding to those different imaging modalities. We like to say that molecular imaging is non-invasive. That's true. We don't actually physically probe the mouse, but on a molecular level, we are changing the mouse whenever we do a molecular imaging study. Mice are under anesthetic, for example, right? We have to maintain their body temperature in unusual ways. So it's something to consider, that we have to be careful with how we handle the mice. Well, if 2 agents cause a problem, if an MRI agent injected at high concentration affects MSOT with its contrast agent, well, why not think about making a dual modality agent? And that's shown here, where there are 3 great examples. These agents tend to be nano-sized, an iron-based agent, another iron-based agent on the left and on the bottom of the screen, and also on the top right, a manganese-based agent. In fact, you can see the contrast change in all of these images. The lower right shows that these investigators were able to also get a CT image from their nanoparticle. So this is a tri-modality experiment with just one contrast agent. Now, certainly there is advantages to a single contrast agent, and I think there can be clever ways of using a single agent for multimodality imaging. So I don't want to sound negative about dual modality agents. However, I do think we have to think of this carefully, or we might run into a trap that we're not going to provide as much value as we think we will. And I'd like to kind of back away from this topic of MSOT and MRI to show an example of why specificity is really important for molecular imaging, and why dual modality agents, we have to think of how we're going to use them to improve the specificity of molecular imaging. So first, anatomical imaging does a great job of detecting a lot of suspicious lesions. So we can detect all the tumors or all the pathological tissues. We also detect non-pathological tissues, but in terms of sensitivity, that's okay. We just want to make sure we detect everything. That's sensitivity. And so we don't really need molecular imaging to improve sensitivity. We're already doing a pretty good job. Now, of course, I would hope molecular imaging can improve things, but it's not absolutely required. But what we really need to do is focus on specificity with molecular imaging. And here's kind of thought experiment, if you will. If we have patients, let's say, that have cancer, well, some patients are going to be very easy to treat, and they're going to be low cost on the left side of this graph. They're going to be patients that are difficult to treat, and they're going to be high cost patients, shown on the right side of this graph. Well, diseases such as cancer and many other diseases are really very serious, and therefore we're willing to pay a high price for healthcare. This is not a political comment or an insurance company comment. It's just a fact that we do invest a lot of money in our lifetime into our health, and so we're willing to pay for healthcare to some extent. And so really the key is to lower the average cost of this health delivery that we're providing to our patients. So the second graph has histogram bars that are lower than the histogram bars on the top graph, indicating that if we could just reduce the whole average cost, yes, that would help to improve healthcare delivery. But really where I think a better impact could be made is if we can identify the non-responders. So if we can identify those patients that will not respond to expensive therapies, such as immunotherapy, or that might need 2 or 3 types of therapies before they find the right therapy, we can take them away from these therapies that are not doing them any good. Well, that's going to save a lot of money and actually help to improve the whole healthcare delivery system. Well, non-responders, in my opinion, can be considered false positive patients. We thought they were going to respond to the drug, but they did not. They were false positive. And we need to reduce false positives to improve specificity. That's the definition of specificity, and you can look it up on Wikipedia. I always have to do that to remind myself. Look it up on Wikipedia. So we need to identify these non-responders with molecular imaging. So can we do that with an MSOT and MRI contrast agent? Well, here's a table showing all combinations of true positives and true negatives, false positives and false negatives. And clearly, if we have one agent that can be detected with MSOT and also with MRI, and the diagnostic imaging comes back from both modalities as being identical, either positive or negative, or perhaps falsely positive or falsely negative, well, then only one agent was really needed. We got the same answer from just one or the other, and we did not need both. Now, that's okay. You know, we could add the second one. It's nice to have some validation. But really, in order to reduce cost, we really only needed one. Well, here's an advantage to a dual modality agent, where if you have a true positive diagnosis, that's going to override the false negative from the other imaging modality. And then the false negative could be from MSOT relative to MRI. It doesn't matter which modality is reporting the false negative. But, you know, a disease like cancer is really important. And so we're going to bias ourselves to go with the true positive result and go ahead and treat that patient, or go ahead and consider that, you know, we got a serious issue here, and not just ignore the negative result. So in this case, both modalities provide value. There's improved sensitivity, and we know we've got some potential problem here. Now, the problem is that if we have a true negative case where the patient would not respond to that drug, or, you know, truly had a suspicious lesion that was not a cancer or something, but the other modality gave a false positive result, well, again, we're going to bias ourselves to look closely at that positive result over the negative result. We can't just let it go and say, well, one of the results was negative, even though there's some, you know, indication that you might actually have cancer after all, or might be treatable. We're not going to do that. You know, we just can't say that to our patients. And so therefore, more tests are always going to be needed if there's at least one false positive result. So therefore, our specificity is unreliable if we have a dual modality agent and we get different answers from these different agents. And so in that regard, I think we have to be careful on how we use dual modality imaging, especially with one contrast agent that is telling us about the same biomarker. Complementary biomarkers are different, but the same biomarker with the same agent falls into this category on this table. And then just to reiterate the difference in detection sensitivity, MRI is challenging. A very high level of agent is needed for detection compared to MSOT. And that can be a major challenge for targeting agents that are trying to identify cell receptors. That's feasible with MSOT, not so feasible with MRI. So again, we have to think very carefully about what biomarker we're going to go after if we're going to be using a dual modality agent. Enzyme activities and reporter genes that use enzymes have some advantages because of fast catalytic activity of the enzyme can turn over a lot of agent for MRI, and pH, hypoxia, and other environmental properties are easier to study with MRI as well. Okay, so in the last two portions of this presentation, I'd like to compare MRI techniques with MSOT techniques. It's really not a case that can these two techniques be complementary, because I've just given an argument that using both modalities to interrogate the same biomarker needs to be done with caution, because it may not provide great value for specificity. And instead, I am actually pointing out that there is a bit of a competition between these two modalities. One may be better than another for certain applications. And that's really what caught my attention back in 2013. So here's an example from Ralph Mason's group. Ralph's been doing a great job with BOLD MRI, and more recently also with MSOT as well. In a case where blood oxygen level dependent MRI measures a change in T2 star when the carrier gas or the anesthetic is changed from 21% oxygen, as shown in the very left of this graph on the left, relative to 100% O2 challenge. And by oxygenating the hemoglobin, turning deoxyhemoglobin into oxyhemoglobin, there's an increase in the T2 star relaxation rate and an increase in the relative signal intensity of the MR image. And so that can be picked up very nicely. By the way, this is actually very high quality data. I was impressed with Ralph's publications. He pays careful attention to detail and gets good quality data. I have to admit that my BOLD MRI results in past years are not nearly as clean as what's shown on this slide, and I perhaps need to do a better job myself then. But it does show that there is some variability in BOLD. Kyle Jones, along with Dr. Radna Venkatesan at the MD Anderson Cancer Center, did a comparative study of different imaging MRI methods on a cervical cancer model. They found that the coefficient of variation of BOLD is actually pretty substantial. There is some variability to this technique. By the way, DCE MRI also has significant variability, and I'll get to that near the end of this presentation, relative to other methods that are perhaps a little more reliable. So there's an issue that BOLD MRI has been challenging to get to work preclinically and especially clinically because of significant variability. It's challenging to make it reliable. How does that compare with MSOT? Well, as I pointed out before, you can measure hemoglobin and deoxyhemoglobin signals, oxy and deoxy signals. And so you can measure a percent oxygen saturation, and there's a small technical considerations with the MSOT. So often this is called percent SO2 or SO2 tagged with MSOT as well. But I think it's becoming a very believable number in the literature through studies that Sarah Bondiek has been doing, and also Ralph Mason has been championing this work. I first heard about this from Ralph a few years ago. I thought it was a very clever idea. And here you can see that the percent SO2, the percentage of hemoglobin that has oxygen, is relatively low when there's only air used, 21% O2 for the mouse imaging. And when an oxygen challenge is introduced, 100% O2, well, then the percent SO2 increases. So this delta SO2 is a very nice biomarker showing how much oxygenation or how much hypoxia is in the tumor. And so in this study by the Bondiek group, comparable to other studies that they've shown, they have a criterion for deciding which pixels are responding or non-responding to this oxygenation challenge. You can see that the amount of tumor area decreases in terms of response before and after applying a vascular disrupting agent. And the graph on the lower right also then shows that, with the drug, there's a decrease in the amount of agent that can respond to this oxygen challenge. So very nice technology that's very similar to BOLD. And while it's difficult to appreciate on this slide, I think the variation of this delta SO2 MSOT is eventually going to be shown to be better than BOLD MRI. Now, that's a provocative statement and it has to be shown experimentally. But I think the data so far seems to indicate that this is a pretty reliable technique with the MSOT. So finally, another comparison can be made with dynamic contrast enhancement imaging. And many of us for many years have developed DCE MRI methods, both preclinically and also in the clinic. And we really haven't quite gotten to a reliable DCE MRI method that is quantitative using pharmacokinetic compartmental modeling for clinical studies. Mouse studies can work under very carefully controlled conditions, but there's a lot of variability in this technique. Just to go through the technique, normal tissues, as shown in the image in the upper left, typically have very nicely formed blood vessels, while tumor vessels tend to be very chaotically organized and very leaky. And so the schematics on the lower left of this slide show that normal tissue does not have much contrast agent that can leak into the tissue microenvironment, while tumor tissue is much more leaky and much more contrast agent can leak into this extracellular, extravascular space. So the MR images on the right just show a flank tumor up on the top of the mouse, showing that there's good contrast uptake. You can calculate pharmacokinetic properties quite regularly with this data. By the way, I've often worked with graduate students making T1-based MRI contrast agents, and then I help them out with their first studies at the scanner, and they're always, almost always amazed that the bladder fills up and gets very bright with their contrast agents. It's very exciting to see this in real time in a dynamic movie. However, I tend to think, great, there's 90% of my research grant money going to the bladder again. But that's just how biology works, and that's okay. At least some gets into the tumor. And this is a case that I'm showing on the slide where Celebrex, which was used as an anti-cancer agent, was applied to this mouse, and 24 hours after that therapy, you can see a necrotic center and lower uptake on the periphery of the mouse tumor. So DCMRI can work. However, there are challenges. It can be difficult to localize the arterial input function that's required for standard DCE MRI quantitative modeling. Often the renal artery is found for an arterial input function, or a vein or artery within the leg for femoral AIF is taken. See, this data is very noisy. Often the vessel is identified by just a few pixels, and if those pixels shift, you're going to have a lot of trouble getting a good, reliable AIF for that data set. And there's enough peristaltic motion or motion in the leg that you can have difficulty getting a good AIF. Also, the data is just doesn't have great signal to noise. You can see even this tumor data has significant noise associated with it, and of course the AIF is very noisy as well. And then finally, MRI. Preclinically is typically collected anywhere from 10 to 30 seconds per image. You can go faster with more sophisticated techniques. In the clinic, very often we're at somewhere between 10 and 30 seconds, typically more on the 30-second range or even 1 minute range, with very slow temporal resolution. High-end clinics can do very fast MR imaging at 5 seconds per image, but that's pretty rare. So MRI is challenging to collect good quality data that has a good AIF, good SNR, and good temporal resolution. Well, those are advantages that MSOT has. And so let's walk through the history of this a little bit with DCE MSOT. First, Daniel Rosansky and Vasilis Metristos published a very nice article in 2016 with a 3D MSOT instrument showing 3 different spots within the volume of the tumor, showing 3 different profiles of dynamic contrast enhancement. If I recall, I think they used indocyanine green. I apologize, I don't remember exactly. But the type of dye wasn't really that critical. It was just a good MSOT dye that could be tracked. And so they classified these patterns in the data as being a healthy major blood vessel, a leaky tumor vessel, and a small blood vessel. So there's information there. Now, perhaps not a quantitative methodology yet, but it shows promise. So then the Bone Deep group showed that in a similar way, by doing DCE MSOT, they can now classify different pixels as being retaining the agent, where the agent signal increases and then stays at a plateau, or a clearing region where the agent clears relatively quickly. And so they can classify different pixels and then look at different distributions of pixels in response to treatment and so on. So semi-quantitative analysis, perhaps not a true quantitative methodology, but they are looking at high and low uptake, and then even regions that are non-enhancing regions. So there's even a third criterion there. So that's nice. To further take this a step farther, the amount of change in signal of ICG from baseline to maximum can be measured. The same research group did that analysis and looked at responding and non-responding pixels, had a criterion for which pixels respond and non-respond, delta ICG being greater than 2 times the standard deviation of the noise, relatively good statistical criterion as shown in the upper left. And so in this case, a vascular disrupting agent, or everybody's favorite vascular disrupting agent in molecular imaging, cumbretistatin A4 phosphate. And so you can see that there is a decrease in the uptake of ICG after applying this drug relative to the vehicle control experiment. So getting a little more quantitative, a little more semi-quantitative perhaps. Finally, Lacey McNally and her research group, I think the study was actually done at the University of Louisville at the time, although then she was at Wake Forest and now has moved to the University of Oklahoma and doing really well there, where she was able to find an arterial input function quantitatively, look at the uptake of a contrast agent that happened to be a targeting agent. So there is the issue that this agent is also binding to a cell receptor in addition to just perfusing into the extracellular tumor space. But by doing a simple pharmacokinetic analysis, they were able to calculate a K in and a K out, and you can see the data fits fairly well. This does include consideration of the arterial input function, just like DCE MRI. There was one assumption made here that the artery that was found within the image was at the same depth from the surface of the mouse as the tumor. And that's a reasonable assumption for this particular study. However, the AIF may not always be at the same depth as the tumor in all studies. So a good initial step to doing quantitative DCE MSOT. Well, we've taken that one step further in our research lab, where we can take a tumor. In this case, we injected Genhance 680 and detected the uptake of that agent in the tumor relative to 900 nanometer MSOT as a control. Very easy to find the arterial input function. After all, we can image hemoglobin, oxy and deoxy hemoglobin. So it's very easy to find a blood vessel. 150 micron resolution is sufficient for finding that one or a few pixels that represent the AIF. So very nice to get a great AIF. And also, again, the 1 second temporal resolution, or in this case, 2 second temporal resolution because of the 2 frequencies, is a great advantage when measuring a very fast changing signal such as the AIF. And in MRI, we don't often have that advantage. So here's a standard paradigm: get the signal, convert to concentration as shown in the lower right, and then do pharmacokinetic analysis just like DCE MRI to get a K-trans or a KEP parameter, just like in MRI. But here's the problem. It's difficult to transform the signal into a concentration because we don't know about variable light fluence within the tissue. We could try modeling that. There are very sophisticated ways of modeling the amount of light penetration into tissue, so that signal can be correlated with concentration. But that's really beyond my skill set to do that kind of modeling of fluence. And so therefore, we had to think of a clever way around this. So we took standard DCE MRI 2-compartmental model theory, married that with the standard model of optoacoustic imaging signals, and we came up with a normalized K-trans that, for lack of a better term, we just call K-app. And by doing that, we were able to show that we can take out the effect of the fluence. This was a simulated study where using all values of fluence that are reasonable, we were able to back calculate what K-app or KEP would be based upon the data that was initially formulated with the true K-app and KEP values that we wanted. And if we didn't avoid this issue of fluence, the data was just incorrect. And that can be seen in the imaging maps, where we get a nice uniform K-app and KEP map, while there's a gradient across the maps if we don't avoid this fluence issue. That's indicative of the depth of penetration, that gradient. And so I'm kind of going through this relatively quickly because there are a lot of technical details, but I would be glad to follow up on this type of DCE MRI or MSOT methodology that can actually give you true quantitative pharmacokinetic parameters. So my colleague Julio Cardenas Rodriguez partnered with Ralph Mason to perform this study. We're following up on this to get a more robust data set. But DCE MSOT can show a decrease in KAP and KEP that has nice small error bars, so we get statistically significant results, which is not the case if we use the methods that do not avoid fluids. And so I'd like to make a provocative statement, which as an MRI person kind of breaks my heart, but I think DCE MSOT can actually replace DCE MRI in the clinic someday for those cases of tumors that are accessible 2 to 3 centimeters from the surface of the body. And, there are challenges to DCE MRI, expensive, large machinery, not always available in all countries. I think DCE MSOT could really have a great impact on healthcare if performed in the right way. My colleague and good friend, Sharia Goyle, along with Jorge de la Cerda, Beryl Schuler, and Katerina Katratsu, along with Julio, we've been performing DCE MRI studies as just a qualitative look at the difference of uptake curves with different tumor types, along with this idea of this oxygen-enhanced MSOT method, all in one shot. We can get the whole data set for a mouse in roughly 45 minutes with good stabilization of the mouse physiology, as I keep mentioning. And so here are some maps showing some initial studies, and we're excited to finish off the study once we're allowed back in our laboratories after the COVID-19 issues have hopefully dissipated, and then we can finally finish off the study. So with that in mind, I'd just like to thank everybody who was involved with this work, including Sharia, Jorge, Katerina, Bill, Su Zhang, and our group has also been involved. Also, I would really like to thank some contributors who provided information for today's presentation. And finally, I'd like to leave you with my email address. Feel free to send me an email, or as Tim said, you can get in touch with ITHERA as well. And these slides can be provided to you. The published material that's shown in these slides will be available. So thank you very much. Tim, back to you.
Thank you very much, Marty. So I'll just say a few words while the questions come in and we sort them out. If you do want to ask a question, as mentioned, you can type it in the question box on the GoTo interface, and we will pass it along in a few moments. I want to say again, thank you to Marty for giving such an interesting and broad presentation on the use of MSOT and the use of MRI. Regarding the clinical component, I will mention a bit of a plug that we're running another webinar next week, which will feature the clinical use of MSOT. So we'll follow up with a link to that later. And now I will stop blathering and move to the questions. Quite a few of them, Marty, so we do have a little bit of time for this. The first question is, you've talked about the stabilization of the mouse. Is there anything that you specifically need to do to stabilize the mouse physiology for MSOT?
Oh yeah, great question. So I'm a big fan of controlling temperature very exactly. And I see other studies where the temperature is a bit more variable, or maybe a little bit lower than physiologically relevant at 37 degrees for a mouse. So the MSOT has a great advantage that the mouse is put into a water tank. And the water tank is a great way of keeping the mouse at a very good temperature. However, we took the extra step to get an SA Instruments system to actually periodically check on mouse temperature. We don't use it for every scan, but we like to ensure that the mouse temperature is always at 37 degrees. And we do have some evidence that older mice in the same water bath temperature might have a slightly different temperature in their body, and so we have to regulate all that. So I really spent a lot of time with temperature in particular. Also, mouse positioning is, I think, really important, but that's pretty easy to do on the MSOT as well. The mouse holder really locks down the mouse very nicely, and of course, the bag system holds the mouse very compactly as well.
Okay, thank you, Marty. I'll just read you the next one. Sorry, they're coming in quite fast. It's scrolling along the screen here. What kind of quality control do you perform for the technique?
Oh yeah, others have done a great job with that. So some really nice literature out there. Again, Sarah Bondic and her group have done some very nice studies, leading efforts to do good QA/QC. Ralph again has done some very nice examples there as well. We do spend some time waiting for the mouse to equilibrate in the water bath to ensure temperature. And then also when we change oxygenation from 21% to 100%, we have a significantly long time. I think it's 8 or 10 minutes to ensure that the whole mouse body has equilibrated. You'd think that in just a few minutes would be enough, but we're not really confident about that. So those are the two keys that come to mind in terms of the physiology in the MSOT.
Okay, and the next question, quite an interesting one, is what do you think will be for the future trends in terms of contrast agents? Do you think there will be both MRI and MSOT dual modality agents?
Yeah, it's a great question. Now, as I pointed out, I think that there are opportunities for a dual modality agent with both modalities. However, it can be challenging to think of what is the ultimate value for that. And just because we can do it doesn't mean it'll provide information that's helpful for physicians or their patients. I think there can be, we just have to be careful. I think, again, MSOT is a great molecular imaging technology, and we're just seeing in the last few years some great examples of detecting enzyme activity, reporter gene imaging, pH dyes as well for MSOT. And so I think that can complement MRI. The techniques that are used typically are diffusion-weighted DCE MRI. There's a way of measuring pH as well. So I think we're at the forefront, or we're at the beginning of this era of seeing how to make complementary MSOT and MRI come together. And so it's a good challenge, a positive challenge to have at this point.
Okay, so kind of similar link question. It is, so you've shown mostly cancer and neuro applications today. Do you think there's other MRI focused applications that you could also do with MSOT?
Oh, yeah, great question. Well, no, certainly MRI does have its advantages. Diffusion-weighted MRI for looking at cellularity in tumors is a great example where at least I can't think of a way to do that with MSOT right now, measuring diffusion rates. And then also, T1 and T2 contrast and how that changes in MRI as a biomarker for disease progression or effects of therapy. There's something there as well, and that is not, there's no equivalent in MSOT. So certainly MRI has its advantages, and I did not focus much on that. I think that's more established. I really did focus on the newer parts of MSOT today. So I don't mean to take anything away from MRI.
Okay. Possibly a controversial question, at least for us as a manufacturer, is what do you see as a primary improvement to MSOT?
That is a great question. I think in general, Tim, you probably get this as well, that putting the mouse in the bag system and putting it in a tank of water at first seems challenging and unusual, and there are rare occasions when we have had a bag rip and a mouse expires during a study. It can happen. We have that happen in MRI too. Mice are fragile. But I think that after some practice, it's really not that bad. And it's not a very challenging method to get the mouse positioned in the water bath at all. So could there be improvements to the mouse handling system? Perhaps, but I think it's just fine the way it is, and the improvements are really going to be incremental. Like we've been using a rat holder for our mice just to hold the mouse head in a certain position. Nice improvement, but I would call that incremental. So to be honest, I really can't think of anything more than that. Oh, I guess another aspect that does come to mind is that I think there are some great improvements being made with the image reconstruction and other algorithms for MSOT that ITHERA is doing, other groups are doing as well. That's beyond me again. I'm not at that level of math. So I don't have an understanding of that field, but I do appreciate that the images look spectacular popping off the screen these days.
Okay. One question regarding BOLD and MSOT is both measure hemoglobin oxygenation. Do you think they really are comparable, or do you think they will both show the similar sensitivity to change?
I do think that they are comparable because they are both measuring effects of hemoglobin or oxygenation of hemoglobin. I specifically did not add told tissue oxygen level dependent MRI to these slides, because I think that can be somewhat different than the percent SO2 or delta SO2 measurements. But I think BOLD is comparable. Now, with both of those in mind, it is good to also consider that measuring the properties of hemoglobin in the blood vessel is not necessarily hypoxia in other parts of the biological system, such as the level of oxygen within a tumor cell is related to the blood vessels, but not exactly the same. And so you have to be careful about how you interpret the data. I can appreciate that. But BOLD and SO2, Delta SO2, yeah, I think they're pretty comparable. And yet those studies have to be done to be shown.
Okay, well, we're coming up to the hour. We do actually have a lot of questions left. A lot of people are interested in your light fluence modeling, so we'll address that separately,
Sure.
or individually offline. So I will just say thank you once again, Marty, for a really great talk, and also thank you to the audience for being here today and attending this webinar. And I hope to see you all in the not too distant future.
Tim, thank you so much.